Welcome friends, welcome to our YouTube channel, and today we will be talking about the development of flower. And in development of flower we will focus on a specific model that is called the ABC model. ABC model determines, or you can say tells about the formation of the floral organ. Okay, before starting the exact journey of the floral development, let us take a glance what are the factors which are responsible for activation or the transcription of all these floral organ identity gene. Okay, so if we talk from beginning, so the flower formation starts from the vegetative part of the plant, and the formation of flower occurs from the shoot apical meristem. But before the transcription factor or the other factors which are associated with the formation of the flower in the shoot apical meristem is initiated with the help of the sunlight. Okay, so when the sunlight fall on the surface of the leaf, and we are talking here about the flower formation in model organism, and that is called as the Arabidopsis thaliana. Okay, so whenever sunlight fall on the leaf of the plant, and you know that the Arabidopsis thaliana is the long day plant, so exposure of light for a long duration initiate the activation of or transcription of the constans gene. You know that the constans genes are transcribed in the leaf of a plant. So let us take an example with the help of a diagram regarding the activation of the constans gene. A lots of question in this section has already been asked in the previous CSIR examination, so it is going to be a very important topic for all those examinations. So let us take a look upon this. Let us understand with the help of a diagram. Suppose this is a plant. And you don't know that the plant have the flowers. Okay, so let us make it quite large, so that okay. So this is a flower, and sunlight is coming from the sun, and it falls on the surface of the leaves. Okay, surface of the leaves. So due to presence of, or due to the exposure of a long long duration of the sunlight, what happens, the constans gene, see your constans gene, transcription of the constans gene occur, and the constans protein is made. The constans protein will made, and due to the presence of the constant protein, it act as a transcription factor for another protein, and that is called as the flowering locus T, and it is represented with the help of, with as a T, FT, and that is called as the flower locus T. Okay, so this flower locus T protein is transcribed in the leaf, and the protein is formed in the leaves, but this protein is translocated toward the shoot apical meristem. Okay, and this translocation is done with the help of the companion cell of the phloem, and whenever this is brought about to the other part of the plant, that is called as the shoot apical meristem of the plant. Okay, now after it reaches to the shoot apical meristem of the plant via this process, now FT is going, okay, and it reaches toward the shoot apical meristem. It reaches to the shoot apical shoot apical meristem. Here in shoot apical meristem, This is a SAM, shoot apical meristem. Okay, after it reaches to the shoot apical meristem, what happens? It induces the transcription of another protein, and that is called as the flowering locus D. This is the FD protein, and this FD protein will lead to the activation of the another gene, and that is called as the leafy. This will induce the activation of leafy gene and AP1. These leafy and AP1 are responsible for the formation of or the transition of the inflorescence into the floral organ. Okay, this will induce the transformation of inflorescence to the floral organ, and under the presence of or in the presence of the leafy and API protein. Okay, under the influence of leafy and AP1 protein, what will happen? This FT plus FD, okay, will lead to the formation of leafy and AP1, and this leafy and AP1 will transcribe the another gene set of the gene, and that is called as the floral organ identity genes. Okay, floral organ identity gene, and this floral organ identity gene is represented as ABC, and it is represented as ABC. So let us take a look at this complete figure, that let us take a recall it. So after the sunlight fall on the surface of the leaves, okay, it transcribes the formation of the constans gene, and after transcription the constant protein is formed. Okay, the constant protein acts as a transcription factor of flowering locus T, and this flowering locus T protein is formed, and this is only in the leaves. After the formation of flowering locus T protein, it is transported toward the shoot apical meristem with the help of the companion cell of the phloem tissue. After it reaches to the shoot apical meristem, what happens? It acts as which one? The FT acts as a transcription factor for another protein, and that is called as the flowering locus D, yani FD protein. This FT concentration of the FT and FD protein induces the another expression of the another set of the protein, and that is called as a leafy and APY. And this leafy and AP1 will induce the expression of the floral organ identity gene, and that is called as the ABC gene. And this ABC gene will be responsible for the formation of the flower. So let us concentrate our discussion on the topic ABC model. So there are certain rules, and before undergoing the certain rules, let us take an account of ABC. What does the ABC means? ABC here represents the class of the gene. ABC class, in ABC class of the genes, each and every word has its different gene expression. Okay, so they are called as the A class gene, B class gene and the C class gene. Okay, so let us break it. A, B, C. A class gene, B class gene, and C class gene. Okay, if we talk about the A class gene, if we talk about A class gene, in A class gene, Aptela 2 is expressed. Okay, Aptela 2 protein is expressed, and if you say about the B class gene, we will the Aptela 3 Aptela 3 protein is formed, and another one is pistillata, and it is represented with pi. This is AP2 and this is AP3. Now if you talk about the C class gene, and it is agamous, and this is represented as a AG. Now in ABC model, okay, so till now we have gone through the A, B and C class of the gene. Before proceeding forward, let us understand about the different organ of the flower and how they are arranged step in a whorl. So before proceeding it, let us take an example of the flower and its arrangement. So if we make a flower, a rough diagram of a flower, we see here this is a pedicel. Now there is a leaf, green like structure before or below the petal, and they are called as the sepal. Now this is the colorful structure. This is the colorful structure and it is called as petal. Okay, and another structure is there. And that is called as, this one is the anther and this is the carpel. Anther is male reproductive organ and carpel is the female reproductive organ. Okay, now SE here we will represent as a AC, petal will be represented as petal PE, and this you can say the anther is the unit, and now it is a stamen. You will represent the stamen as SA and carpel will be represented as SA. So this is a flower diagram. Okay, so right now we have the four layer of the flower, or you can say the four whorl of the flower. First one is the sepal, which is lying at the basal part of the petal, and before and after the sepal there is another colorful part of the plant that is called as the petal, and thereafter the male reproducing organ that is called the stamen, and thereafter the female reproducing organ that is called as the carpel. Okay, so now if we look at just as a circle like diagram, then we will see that suppose 1 2 3 and 4. Okay, so this is the first whorl, second whorl, and this is third whorl, and this one is the fourth whorl. First, second, third and fourth. Now let us take an example. The first word is which one? That is the sepal one. Here, suppose this is the sepal. The next one is the petal. Third one is the stamen. And fourth one is the carpel. So, at the inner part, the carpel, right, which represents the female reproducing organ. At the third part, that is called the stamen, male reproducing organ. Second part one is, you can say, the petal, the colorful part of the plant, and the lower is the green colored sepal. So, till now we have gone through the structure of a flower, which are arranged in the four words: 1 sepal, 2 petal, 3 stamen, and 4 carpel. Now let us try to understand what are the arrangement of the gene, what are the basic arrangement of the gene which is expressed in the plant flower. Okay, so let us understand all these things. Now what happens here, let us make a diagram here. You can say this is the arrangement of the flower. Now ABC model which we are talking about, okay, so this is a set of the B gene, and now this is the set of the A gene, A, and this is the C. Now what will happen thereafter if you're talking about? Okay, first A gene, only A gene is responsible for the formation of the sepal. It will be responsible for the formation of sepal, which is encoded by AP2. Okay, now clear. AB is responsible for the formation of the petal. Okay, and this AB will be encoded by first AP2, and if you recall the previous thing which we have discussed, AP2, then AP3 and PI. Okay, so this is A class gene and this is a B class gene. Okay, now it is AB. Clear hai? Now BC will give rise to the stamen. Okay, and this is represented with the help of the B class gene, and that is called as the AP3 and PI, and the C1 is agamous, that is represented by AG. Now another set of the gene is the class C gene, and this class C gene is responsible for the formation of the female reproducing organ, and that is called as the carpel. And the A class gene, C class gene only mean for agamous, that is AG. So this is the basic arrangement for the formation of the flower, and based on this we will try to understand how the part of the part of the flower is formed. So let us take an example here. Till now we have understood these are the factors, these are the possible set of the arrangement which will be responsible for the formation of the flower. If you will ask that why didn't we have written, why didn't we have written the AC? AC, the most important thing is the A and C are antagonist. A and C are antagonist to each other. Means if A is not expressed, then the C will be dominant there. Means the C will be dominant here, and the part of the plant in absence of A will be formed at the place of the A. Okay, suppose A is not expressed or it is mutated. Okay, the part of the flower which were formed with the help of the C gene, that is called as the carpel, and in the absence of A, at the place of A, the carpel will be formed. Okay, so let us take a one by one. Thus we will try to mutate first single gene, then we will try to mutate the double gene, and then after we will try to mutate the triple gene. Then after we will take a summary of the individual genes by name of the genes, and we will see what happens, what could be the possible consequences after the mutation of the single, double and the triple gene. Okay, so let us take an example. First of all, we will consider this figure as a wild. Suppose take this as a wild. Suppose this is wild and no mutation occurs here. Okay, now first let us take an example of A mutated, means A is not there, means the class A gene is completely vanished. Okay, so first make a forward 1, 2, 3 and 4. Now let us understand, suppose there is no B, again 1, 2, 3, 4. Suppose there is no C, the class C gene is absent. Again we will try to make suppose AB is absent. AB is absent. Then 1, 2, 3, 4. Okay. Now, so it is full one. Now let us understand another one. Suppose AB is absent, then BC is absent. Okay. Now again 1, 2, 3 and 4. If we think that now the AC is absent, AC is absent, okay, or AC is absent, again we will try to make 2, 3 and 4. Now suppose all those three genes A, B, C is absent, then what will happen? Now again let us make 2, 3 and 4. So we have 1, 2, 3, 4, 5, 6 and 7 fate. Now let us understand here one by one. Suppose this is a wild variety, means there is no mutation occurs. Okay, suppose if A mutation occurs, means the class A gene, there is a mutation in the class A gene, then what will happen? Okay, so let us take an example. Let us take an example. Suppose what will happen if the class A gene is mutated. Okay, so let us make a diagram which we had already made. Okay, no, let us also make a diagram here for the wild variety. Now here B, A, C, C. Okay, here sepal was being made, here the petal was there, here the stamen was there, and here the carpel was there. Okay, now here what will happen in case of the ablation, means the A gene is absent, A gene is absent. So in previous discussion we have told you that A is antagonist to the C, means A is absent, the C will be expressed in place of the A. So here is a place of A, but it is mutated. Okay, then what will happen if it is mutated, then what will happen? It will be occupied by, here the C and C is occupied as usual. C and C is occupied as usual. Now what will happen? Now the A, in place of A, the C will be expressed. Okay, the C will be expressed. So let us take, let us match what will be the fate. C, carpel will be there. Now again the carpel will be there. Already we have told you the C is antagonist to A, means in absence of the A, the C will be expressed. Now what will happen thereafter? So what will happen there? Okay, now this B and C is expressed by the stamen. Right now we don't have A. So what will be the fate of all this? B and C, means again here the only stamen will be formed. Hope it is correct. Okay, so what will happen? Let's take in carpel, stamen. Hope you are correct. Hope you get the things clearly. Now let us move toward if the B is absent. Okay, if B is absent means A and C is expressed here. A is expressed here, but B and C are not expressed. B and C are not expressed. Then what will happen? Now first only sepal will be formed as usually like the wild one, and again the carpel will be formed. Now what will happen if B is not present there? Okay, if B is not present there, then what will happen? Completely B means there will be no formation of what? The B class of gene represents AP3 and pistillata, which is not there. So this will be occupied by an induction. A will induce formation of the sepal laterally and C will induce the formation of carpel laterally. Okay, then what will happen? Only A is there. Only A is there. So what will be the formation of this part? Sepal, only because only A is there and A is responsible for the formation of the sepal. So sepal will be formed, and in case of the C, only C is there, and you know that the C is responsible for the formation of the carpel. So here is the arrangement of sepal, carpel. Now at the mid, you know that is the carpel, and again after mid, that is called as the carpel, and the first wall will be the sepal and the second wall will be the sepal. So the arrangement will be like this: sepal, carpel. Okay, now let us take an example. If what will happen if the C class gene is absent? What will happen if class C gene is absent? So as you know that A and C are antagonists. If C is absent, then the A will be expressed at the place of the C. A will be expressed at the place of the C. So B is intact here. B is expressing. A is also expressing, but the C is absent there. Okay. So at the place of the C, A will be expressed. Okay. So let us take an example. A and A. You know, the A will be responsible for the formation of the sepal, and again A will be responsible for the formation of sepal. Now let us take AB. What is the function of AB? AB. That is the petal, and this is the petal. Okay. So the arrangement will be sepal, petal, and petal. This is very quite simple. Now let us move toward the double mutation. If we move toward the double mutation, then what will happen? If AB is absent, okay, let us make another diagram. Okay, A, B, this one is absent. Okay, so first we know if B is absent, then B is absent here. Okay, just remove it. B is absent, and you know that this place is occupied by B and A and C. Suppose A and B is absent, B is absent, and again A is absent. You know that A and C are antagonists, then the C will be expressed at the place of A, and then what will happen? The only entire part will be the carpel and carpel, carpel and carpel, 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 carpel. So entire word will be made up of the female reproductive organ, only female reproductive organ. In place of sepal there will be carpel, in the place of the petal there will be carpel, in place of the anther you can say stamen there will be carpel, and in the place of the carpel there will be a carpel. So the entire individual will be the carpel. What will happen if AB is absent? Then after let us take a look toward the BC. Okay, so if the BC is absent, means C is absent, means A will be expressed at the place of the BC, at the place of the C. If BC is absent, then you know that C is absent, then A will be expressed at the place of the C. Okay, A and A, and now this is not here, minus B is there, means B is not expressed here. So what will happen? All will be sepal, 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 sepal and sepal. That's it. Sepal, sepal, sepal and sepal. Finished. Okay, A, B, C. Okay, what will happen if there is an absent of AC? What will happen if there is an absent of AC? Okay, if AC is absent, A, C, C, B, B. If AC is absent, then what will happen? So this is the undefined condition, and it will make a hybrid. Okay, it will make a hybrid of what will happen here. If AC is absent, A and C is absent, means only B is there, and you know that B is involved in the formation of petal and stamen. Okay, it will be in the formation of the petal and stamen. So this will give rise to the petal stamen hybrid. So it will make a petal stamen hybrid. Petal stamen hybrid. So this is about the structure of in case of AC mutation. Now what will happen if all those three genes is mutated? Okay, if all three genes is mutated, then what will happen? It will look like a leaf-like structure, means only leafy will be expressed. Okay, means only leaf-like structure in all four world of the plant will be expressed, means the plant will look like leaf structure. Okay, so a lot of the question which has already been asked in the CSIR NET examination, and all these questions, okay, keep on rotating within this case study. So it is very useful. Now let us understand these things by making mutation in enzymes. Suppose we were talking about till now, we were talking about the mutation in case of the class A gene, class B gene. The question is also asked if we mutate one gene. A lot of the question is asked. So if we come to know that if AP2 is ablated or mutated, then what will happen? Let us take an example. Okay, so AP2 is represented by A class gene. Okay, A class gene. The question will be asked, if what will happen if AP2 gene is mutated, what could be the fate of the flower? So let us again make an photograph of this thing. What will happen if AP2 is mutated? So AP2 is mutated, then what will happen? You know that the AP2 is a class A gene, so here only the C will be expressed and then here B will be expressed. A will be replaced by the C. So now what will happen here? Carpel, BNC, and that is called as the AC stamen. So this will be the fate or outcome of the floral arrangement. Okay, so in this way a lot of the question will be asked. Suppose you will be asked that if agamous is absent, then what will happen? If agamous is absent, then you come to know that the agamous is expressed in the class C gene. Okay, if class C gene is not there, then what will happen? A will be expressed at all these places. And the resultant outcome will be sepal, A and B petal, the petal. The arrangement will be like this. Okay, the arrangement will be sepal, petal, sepal, petal, and sepal. So in this way we come to understand how the floral arrangement and the organ of the flower is produced on the flower and how they are maintained. Okay, so this was about the ABC model. Hope you like the session, and if you like this session, then don't forget to subscribe, like and share. Thank you for watching, and keep watching the videos.